Hello students, a warm welcome to all of you in ITLS Academy. In today's lecture, we will discuss about the DNA sequence analysis. In today's lecture, we will discuss how we will analyze the DNA sequence. Okay, so we start today's lecture. First, we understand what is a DNA sequence analysis. What is a DNA sequence analysis? DNA sequence analysis is the process of examining and interpreting the nucleotide sequence in DNA to understand its structure, functional, and evolutionary relationship. Okay, what is a DNA sequence analysis? DNA sequence analysis is basically a type of process. Okay, in which we identify the sequence of DNA. In sequence, basically we identify the sequence of nucleotide. Nucleotide in DNA. In DNA, there are four types of nucleotide sequences present. First is a adenine. Second is a guanine. Third is a cytosine. And fourth is a thymine. To find out the nucleotide sequence in DNA, the process by which we identify the nucleotide sequence in DNA is known as a DNA sequence analysis. Okay, why we will perform this DNA sequence analysis? We perform this DNA sequence analysis because we want to understand the structure, functional, and structure of DNA, function of DNA, and evolutionary relationship. Okay, DNA analysis is the process to understand the determine and exact order of nucleotide sequence. Okay, nucleotide sequence in DNA molecules. There are four types of nucleotide sequence: adenine, cytosine, guanine, and thymine. Okay, this information is crucial for understanding the genetic code and is widely used in the area of genetics, medical medicines, biotechnology, and evolutionary biology. Okay, to analyze the nucleotide sequence is very important for what? For understanding the genetic code. What is a genetic code? Basically, genetic codes are the sequence, basically three sequence of nucleotide. For example, UAA. What is a UAA? That is a uracil, adenine, and adenine. This is a genetic code, and this is responsible for the stop or terminate the. It is a stop codon. Okay, and this also responsible. These genetic codes is also responsible for the translation process. For example, AUG. AUG is a genetic code that code for the methylene. What is a methylene? Methylene is a type of methylene is a type of amino acid, and these amino acid is the building block of protein. That's why this DNA sequence analysis is very important. Okay, and this DNA sequence analysis plays a very important role in genetics, in medicines, in biotechnology, and in evolutionary biology. Okay, DNA sequence analysis is a crucial process in understanding the genetic codon, identify the variation in genomes, and study their function and evolutionary significance. Okay, it involves a range of computational and experimental techniques used to identify gene, regulatory element, mutation, and evolutionary pattern. Okay, for analysis of DNA sequence, there are lots of techniques are involved. Okay, the result of DNA sequence analysis are crucial for various fields such as genomics, biotechnology, medicine, evolutionary biology, life science, etc. Okay, this information can be used to study the wide range of biological process for understanding the genetic basis of disease to track the evolutionary relationship between the species. Now we understand the basic step of DNA sequence analysis. First step is a sample preparation. Okay, first step is a sample preparation. In this step, we extract the DNA. Extraction of DNA. We extract the DNA from different sources such as blood, cell, hair, mucus, etc. First, we extract the DNA from these biological samples. Okay. After there are lots of method also available to extract the DNA. Okay. After extracting the DNA, then we prepare a library. Okay. First, after extraction, we perform the PCR. Why we perform PCR? To amplify the DNA. In PCR process, we increase the number of DNA. After performing the PCR, then we prepare the library. Okay, fragmented DNA, adding adapter and preparing for its sequencing. Okay, after preparation the DNA sample for sequencing, then we perform the sequencing process. Okay, next is the DNA sequencing. Okay, DNA sequencing is a process by which we analyze the DNA sequence. In DNA sequence analysis, to obtain the actual DNA sequence, this this involves using the DNA sequence technologies. What is the role of DNA sequencing? DNA sequencing is a process by which we analyze the exact DNA sequences. Okay, there are lots of DNA sequencing methods are available in the field of life science. First is a Sanger sequencing, next is a next generation sequencing, third is a Max Gilbert sequencing, pyro sequencing. These are the some method by which we easily perform the sequencing method. Okay. In Sanger sequencing, Sanger sequencing is the traditional method that use the chain termination nucleotide to produce the sequence of very length. Okay. Next is the next generation sequencing. Modern high throughput technology that can sequence the millions of DNA fragments simultaneously offer the great speed and lower cost compared to the Sanger sequencing. This next generation sequencing method is very advanced method. It is a very advanced method. By the help of this sequencing, we sequence the millions of DNA fragment in very short time duration. Very short time duration. Okay, next. Once we sequence the DNA sequence, then we store this DNA sequence in the databases like GenBank or ENSEMBL. Okay. First, first we isolate the DNA sample. We isolate the DNA sample. Then perform PCR. Then prepare libraries. Then perform sequencing. There are lots of methods are available to perform the sequencing. First is a Sanger sequencing, then Max Gilbert, then Pyro, then next generation sequencing. After performing the sequencing, we analyze the DNA nucleotide order in DNA structure. Nucleotide order in DNA structure. After analyzing the nucleotide sequence, we store these information into different data source. different data source or data bases. For example, GenBank and ENSE, EMBL. Okay. Now we discuss the data analysis. After performing the sequencing, we analyze the data. Okay. For analyzing the data, first is a quality control, then assembly, then annotation, then variant changing, variant calling, then variant calling, then comparative genomics. Okay, first we understand the quality control. Quality control assessing quality control in a quality control we assessing the accuracy and completeness of the sequence data. In quality control we assessing the accuracy and completeness of the DNA sequence. In assembly, we reconstruct the original DNA sequence from the fragmented reads. Annotation, we identify the gene, regulatory element and other biological features within the sequence. Variant calling, detecting the genetic variation such as SNPs, that is a single nucleotide polymorphism, indels and structure variant. Next, comparative genomics. Compare the DNA sequence across the species to identify the conserved region and understand the evolutionary relationship. Okay, in data analysis, first we analyze the quality of the DNA sequence. First we analyze the quality. In quality, we analyze the accuracy and completeness of DNA sequencing data. Next, we assemble or reconstruct the original DNA sequence. Okay, after analyzing the DNA sequencing, we find out the order. Okay, when after finding these order, we construct the whole double stranded DNA with this fragmented sorry hmm. After checking the quality, we assemble or reconstruct the original sequence of data from the fragmented read. Okay, after reconstruction the original DNA sequence, we analyze the gene, regulatory elements and other biological features and elements within a DNA sequence. After identify the gene, regulatory elements, then we identify the genetic variations such as mutation, single nucleotide polymorphisms, and structure variant, etc. After completing this whole process, we compare these nucleotide sequence from the DNA sequence of other species to identify the evolutionary relationship. Okay, this is the data analysis. Okay, after analysis the DNA data, after analyzing After analysis the data, we perform other important tasks. First is a sequence alignment. Why we perform sequence alignment? We perform the sequence alignment to identify the similarity between the sequences. Okay. Sequence alignment is a fundamental step in DNA sequencing analysis. It involves the comparison of DNA sequence to identify the region of similarity that may indicate the functional, structural, and evolutionary relationship. Okay, this sequence alignment is a fundamental process. Why we perform this sequence alignment? To identify the functional, structural, and evolutionary relationship. There are two types of sequence alignment. First is a pairwise sequence alignment, second is a multiple sequence alignment. Whenever we compare the two DNA sequences, then this alignment is known as a pairwise sequence alignment. Whenever we compare the more than two sequences of DNA, then that is known as a multiple sequence alignment. Okay, basically to perform the alignment we use BLAST. Okay, what is a BLAST? BLAST is a basic local alignment search tool. These are very commonly used alignment tools. This BLAST helps to find the homologous sequence in large database and used for gene identification, evolutionary relationship, and functional analysis. Okay, first, sequence alignment is a fundamental step in DNA sequence. Why we perform sequence alignment? Because we want to find out the functional, structural, and evolutionary relationship between the species. There are two types of alignment method. First is a pairwise sequence alignment, second is a multiple sequence alignment. Okay, so basically to perform the alignment tool we use BLAST. What is a BLAST? BLAST is a basic local alignment search tool. This tool is very helpful to identify the gene identification, evolutionary study, and functional analysis. Okay, basically to perform the pairwise sequence alignment, we use emboss. Emboss is also a tool that are used to perform pairwise sequence alignment. Next, cluster omega. This cluster omega is also a tool that is very helpful to understand the to perform the multiple sequence. Okay, next. Now we understand the gene prediction. Okay, identify the gene within a raw sequence is essential for understanding the functional element of the genome. Okay, if we find out the whole genome and sequence of any DNA, then this is very important to find out the functional region of this genome. This functional region is also known as a gene. This gene is also responsible for the translation of protein. Okay, to perform the central dogma, this gene plays a most important role. In bioinformatics, there are lots of algorithms available to perform to find out the gene. In gene prediction algorithm, we are looking for the open reading frame. In open reading frame, start and stop codon is present, and other patterns also present that are essential for the performing the transcription and translation process. Okay, now we understand the method that are used to perform the gene prediction. First is a ab initio. First is a ab initio gene prediction. Okay, algorithm like GenScan used to model the gene structure to predict the gene without external data. This gene bank is a tool that are used to predict the gene. Okay, this is a type of ab initio method. Next is a homology based gene prediction. Okay, relies on a known gene from related to the organism to predict the gene based on a sequence similarity. This is a direct method to perform the ab initio gene prediction which does not require any external data. But in homology modeling, in homology based gene prediction, here we require the similarity sequence. And after okay, and we compare the similarity sequence with our here we in homology based modeling we required a query sequence, and these query sequence and these in homology based in homology based gene prediction we required a query sequence, and this query sequence have some similarity with our target sequence. Okay, due to this similarity we identify the gene. Okay, relies on known gene from related organism to predict the gene based on a sequence similarities. Okay, if our prediction is based on this similarity, then this method is known as a homology based gene prediction. In ab initio gene prediction, we does not require any query sequence. Okay, but in homology based gene prediction, we require a query sequence that have a similarity with our target molecule. Okay, now we discuss the motif and regulatory element detection. Next and most important point is to find out the regulatory and conserved region in the gene or genome. Okay, what is a regulatory element? Regulatory elements such as promoter, enhancer, silencer, transcription factor, transcription finding, transcription factor winding sites, control. Okay, promoter, what is the role of promoter? To enhance the rate of transcription. Enhancer also enhance the rate of transcription. Silencer will decrease the rate of transcription. Transcription factor winding sites, these are the sites where transcription factors are bind. Okay, and these elements are essential to to regulate the gene expression. Okay, next, motifs. Motif discovery tools like MEME or FIMO are used to identify the conserved pattern in DNA sequence that may serve as a regulatory element. Okay, these are the bioinformatic tools that are used to identify the conserved pattern in DNA sequences. Okay, next, variant analysis. Single nucleotide polymorphism, that is a SNP, insertion, deletion and other variation in DNA sequence can affect the gene functions and lead to disease. Identify these variation is a critical in medical genetics and personalized medicine. Okay, SNP, that is a single nucleotide polymorphism and mutation. Okay, mutation may be due to mutation due to insertion due to deletion due to frame shift due to chemical method due to physical methods. These all variants are very important because they directly affect the function of gene. Affect the function of gene. Due to the mutation, there are a lots of this mutation, and SNP is the reason of lots of genetic diseases. Okay, this plays a most important role in medical genetics and personalized medicines. Okay, variant calling algorithm like GATK, that is a genome analysis toolkit. This tool is used to identify and to detect and classify the variant form from genomic sequence. This tool is used to identify the variant. Okay, next is a functional annotation. Functional annotation tools like ANOVAR. These are used to predict the impact of variant on gene function. This ANOVAR is used to find out the impact of variant. This is used to identify the variant, to detect and classify the variant, and this tool is used to identify the impact of variant on gene function. Okay, now we discuss the next, that is a phylogenetic analysis. This phylogenetic analysis is very important to find out the evolutionary relationship. Evolutionary relationship between the species. Okay, by the help of phylogenetic tree, scientists track their evolutionary history. For reconstruction, for the construction of phylogenetic tree, there are lots of algorithms available in bioinformatics, such as UPGMA method. It is a type of distance metric. Next, neighborhood joining method. Next, maximum likelihood methods. These are the algorithms which are used to construct the phylogenetic tree. Next is a MEGA. MEGA 11 is a tool that are used to reconstruct the phylogenetic tree. The full form of MEGA is Molecular Evolutionary Genetic Analysis. Next is a RAXML, RAXML. This is also this is also a tool that are used to construct and analysis the phylogenetic tree. First is MEGA. Next is a RAXML. These are the tools which are used to construct the phylogenetic tree. UPGMA, maximum likelihood and never joining. These are the algorithms which are used to construct the phylogenetic tool. These are the algorithm and these are the tools. this never joining, maximum hood, and UPGMA. This is the algorithm and this is the tool. Next is a comparative genomics. This plays a most important role in the field of biotechnology, in a field of medicine, in a field of evolutionary biology, etc. By the help of comparative genomes, we identify the conserved element such as gene, regulatory region, and chromosomal structure. By comparing the genome, researchers gain insight in the evolutionary process and functional elements. Okay, first is the synteny. Synteny analyze and help to detect the conservation of gene order across the species. This synteny is used to identify and for the detection of conserved gene order. Synteny is used to detect the conservation of gene order across the species. Next is the orthologous gene identification. Orthologous gene identification also plays a most important role in the comparative genomics. What is an orthologous gene? Orthologous gene in different species derived from the common ancestor. This orthologous gene identification plays a most important role to construct the phylogenetic tree. For the study the evolutionary relationship. Next is a functional annotation. Functional annotation plays a most important role. Once the gene and other elements has been identified, then they need to be annotated with functional information. This involves linking of DNA sequence of biological function, such as predicting the function of gene product or identify the associated pathway. For detection the functional annotation, we use the UniProt and KEGG and Geo data sources. Okay. Whenever we identify the gene, then we annotate their biological function. Okay. And in bioinformatics, UniProt, KEGG and Geo, these are the databases present which are responsible for the identification of functional annotation of gene and proteins. Okay, basically now we discuss the bioinformatic tools and techniques for analysis the DNA sequence. First is a BLAST. BLAST is used to identify the similarity between the DNA sequences. Next is a Cluster Omega. This tool is used to perform the multiple sequence alignment. Next is a gene prediction tool. GenScan and Agostus is used to identify the gene. Next is variant calling and SNP analysis. Genomic analysis toolkit is used to detect the SNP and variant in the DNA sequence. Next is a BCF, that is a variant calling format, a file format used to store the information about the variant. This tool is helpful for is storing the information, and this is tool is used to for the detection. Next tool is a phylogenetic tool. Phylogenetic tool, that is a Megha and Rexmal. These are the tools which are used to construct the phylogenetic tool. Next, functional annotation tool. These are the interproscan and gene anatomy is tools or databases which are used to find out the functional annotation in gene. Next is a motif finding tool, that is MEME and FIMO. MEME is multiple EM for motif elicitation, that is used to identify the motif or conserve reason in biological sequences. Next is a FIMO, that is a find individual motif occurrence. Next is the FIMO, that is a find individual motif occurrences. Okay, this tool is used to search for occurrence of known motif in DNA sequences. These are the bioinformatic tools that are used for analysis the DNA sequences. Next is a genomic browsers. First is a USCS genome browser. It is an interactive platform for viewing the genomic data such as gene annotation, regulatory element, and comparative genomic data. Next is a ENS-EMBL. This provides the access to annotated gene and tool for exploring comparative genomics and evolutionary relationship. Okay, now we discuss the application of DNA sequence analysis. First, in disease research. By the help of, whenever we identify the DNA sequence, we easily identify the genetic variant that are associated with disease. Next, we easily find out the potential of therapeutic target by the analyzing disease associated genes. Next, evolutionary study. By the help of we easily investigate the evolutionary relationship between the species. Next, personalized medicine. Analyzing the individual genetic variation to tailor and medical treatment based on a patient's genetic profile. Predicting the DNA drug response, drug risk, identify the biomarkers for precious therapies. Next, in forensic science, DNA sequence analysis is used in forensic investigation to identify the individual based on a genetic evidences such as a paternity testing or crime scene analysis. Agriculture and biotechnology, in crop and livestock improvement, the DNA analysis is used to identify the gene related to traits like disease resistance, growth rate, and nutritional content. Next, genetic engineering often relies on understanding gene sequence to introduce the beneficial trait in plants and animals to produce the GMO, that is a genetic modified organisms. Okay, this next application in genomics, in transcriptomics, in proteomics, in metagenomics, in personalized medicine, in forensic science, evolutionary biology, drug discovery. Next, we discuss the challenges and future direction. First, in data analysis. Next, data privacy. Then, ethical consideration. Then, technological advancement. In data analysis, the share of first data analysis, the share of volume to generated by the next generation sequencing poses a challenge for analysis and interpretation. Next, data privacy protection. Protecting the privacy of genetic data is growing concern. Next, ethical consideration. The use of DNA sequence data raises the ethical questions such as the genetic discrimination and informed consent. Next, technological advancement. Continued advancement in sequencing technology will enable even deeper and comprehensive analysis of DNA. Whole genomic sequence. With decreasing the cost of sequencing, whole genome sequencing is become a more feasible for analysis the complete genomic rather than just a specific region. CRISPR technology. DNA sequencing analysis aids in identify the target sequences for genome editing using the CRISPR allow the precious genetic modification. Next, single cell genomics. Analyze the DNA at the level of individual DNA, provide an insight into the cellular heterogeneity, and can be used in cancer research and developmental biotechnology. This is the challenges and future direction for the DNA sequence analysis. This is all about today's lecture. Okay. Please follow ITLS Academy on Facebook, Instagram, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share and subscribe ITLS Academy. Thank you so much.